Welcome to Biology Made Easy. Today we are explaining the structure of DNA molecule. Welcome. DNA is nucleic acid, because it's found in the nucleus. And then it's a polymer of nucleotides, a huge molecule with repeating units, and the units are nucleotides. So we want to look at the components of DNA, and that means the components of a nucleotide. In a nucleotide, you have nitrogen-containing bases. This gives the basic nature to DNA. And the nitrogen-containing bases, you have purines. These are 2 rings. And you have pyrimidines, 1 ring. Purines, you have adenine, guanine. Pyrimidines, you have thymine, cytosine. You also have uracil, but it's in RNA. That's why I ran a pen through it. Let's not worry ourselves about the chemistry of it now. We'll talk a bit about it as an appendix in this lesson. Then phosphoric acid as one of the components. And this is the structural formula of phosphoric acid. Then we have pentose, 5-carbon sugar. It means in DNA, this oxygen on carbon 2 is missing. That is why it's deoxyribose. We have labeled the position of the carbons. This is carbon 1, 2, 3, 4, 5. When I have drawn it big here, this is the deoxyribose sugar. This is position of carbon 1, carbon 2, carbon 3, carbon 4, and that is carbon 5. Now, we will look at briefly how the bonding of these 3, the nitrogenous base, phosphoric acid, and the deoxyribose sugar produces a nucleotide. At the end, as an appendix, we'll explain the chemistry well. But for this time, we want to say that the phosphate bonds here on carbon 5. And then there's also bonding here with the nitrogenous base. When there is such a bonding, you end up having at carbon 1 here, you end up having the base, nitrogenous base is here. And then, so we have carbon 1, carbon 2, carbon 3, 4, and carbon 5 is here. And on carbon 5, you have the phosphate. So, this is how the bonding goes to produce the nucleotide. So this is a nucleotide. How the nucleotides link to form the DNA molecule. The sugar and the phosphate, here is phosphate, sugar, form a long unbranching too, with the bases taken at right angles. So we are going to demonstrate how it happens. The 2 chains are anti-parallel. I have a nucleotide here. This is the phosphate, this is the sugar, this is the base, and this base is adenine. So the anti-parallel nature goes like this. You have this nucleotide, the other nucleotide in the other chain is turned upside down like this. Phosphate always attaches on carbon 5, the base always attaches on carbon 1. So you have this one here. The other chain is just a turnaround where the phosphate goes down, still attached to carbon 5. All right? So we go back to this chain. We have adenine with thymine. Another sugar is linked at this end where you have carbon 3 linking here. We'll talk about this bond. You have one linked up in the same way. So you have this chain here, and as I said, this other chain that produces the antiparallel is just a turnaround opposite. So you see cytosine is here, carbon 3 here will join with carbon 5, and here we go. See how they link? Adenine will go with thymine. So this is what produces the antiparallel nature, and we started this at 3 prime, and you see we are ending on 5 prime, and this chain we started on 5 prime here, and we are ending on 3 prime. This is what produces the antiparallel nature. You see that you have carbon 5 linked with the phosphate, and it's going to be linked with carbon 3. That is how it goes. The link between these bonds, carbon 5, carbon 3, this bond, the two sugars, linking the two sugars through the phosphate, this link is the same all the time. This link is known as phosphodiester bond. It's the bond that links successive sugars of nucleotide in the DNA or RNA. So you have 5 linking to 3, 5 linking to 3, 5 linking to 3. This is a 3 prime and it ends at 5 prime. So the bases that stick at right angles of the sugar is always attached on carbon 1, note carbon 1, carbon 1, carbon 1, carbon. And the bases that are complementary bases, you have adenine always pairing with thymine, and hydrogen bonds. Put the two chains together at the bases here, and you have 2 hydrogen bonds. Cytosine and guanine has 3 hydrogen bonds holding them together, and that is how it goes. Hydrogen bonds, hydrogen bonds. Then we also have to note that each of these chains twist up in a helix. So this chain will twist up in a helix, this one too will twist up in a helix, and we end up with a double helix structure of DNA. Everything is constant with the DNA molecule. You have constant measures of DNA molecule. And the first one we talk about is that the phosphate is always attached on carbon 5, and there's a phosphodiester bond always between carbon 5 and 3. Besides that, the distance between the two chains, this chain and this chain, is 2 nanometers. Then the distance between these two base pairs is also always constant at 0.34 nanometers. And then the double helix, every one complete turn of the double helix has about 10 base pairs in it. And the length of the turn is 3.4 nanometers. The DNA double helix is coiled around histone proteins. Sort of the histone proteins serves as a support and is coiled around these histone proteins. Not only that, the DNA molecule is further coiled so much, that is the tertiary supercoiling of the double helix. This double helix recoils on itself. So, like in human beings, eukaryotes, you have 2 meter length of DNA fills a nucleus of 0.6 raised to the power minus 5 meter nucleus. So the nucleus is so small, and so the DNA is coiled, double helix, feather coil, and it uses protein histone to give it support. And all these have constant measures. It has a whole lesson on DNA packaging. Well, we'll end it here with the structure. And as I said, the appendix, we want to look at a bit of chemistry. Good. Yes, so the chemistry, here is phosphoric acid. Here is deoxyribose sugar. The phosphoric acid links at carbon 5 on the deoxyribose sugar. So the link is here, and this water is removed. H2O, water is out. And so you end up with this. So this is the sugar and phosphate. This is how we come. This is the result of it. The sugar and the phosphate, that's the link. Then we have the link between the nitrogenous base and the sugar is at carbon 1. So we have this condensation reaction, water is removed. And you end up with a link like this. Simple. Then we put the two together and we have our nucleotide here. So this one and this one, we have our nucleotide. The phosphate is on carbon 5. The nitrogenous base is linked to carbon 1. Simple. So if we've been helpful, give us some and please subscribe. Thank you very much and goodbye.